right, I'd like to thank you guys for being here. I'm Jennifer Dever from the University of San Francisco, and I'm going to talk about Rana boylii and a small-scale population genetics study I've been working on for the past couple of years. So in this talk, basically, I'm going to introduce the hypothesis that I tested, specifically the methods, both field and molecular, and results, conclusions, and hopefully future work. So why study Rana boylii, the foothill yellow-legged frog? You've already heard a lot about Rana boylii this morning, and so the conservation implications should be somewhat obvious. The most interesting thing about this species to me is that it is riparian, thus it's limited to rivers and streams. Because of this restriction, this may affect its dispersal in that its movement patterns are limited to the river corridor or the stream-river connection, as opposed to overland. So that could have conservation implications because of their dependence on the quality of the river habitat. But again, as you also know, as it says here, they've experienced a severe decline, and they are listed as a state species of special concern. So we need to understand what is behind this decline. So far, Rana boylii has been competing against introduced bullfrogs. There has been records of predation by crayfish, exotic crayfish, and of course also habitat destruction has been implicated. I have been collecting individuals from various streams that are tributaries of a section of the Eel River located in Mendocino National Forest and adjacent to it. This is a typical stream that we find these guys on, and here's just a picture, dorsal surface, and then of course the yellow legs. My students and I have been collecting these frogs now for a couple of years because I'm interested in the conservation genetics aspect of the species. And what I'm going to talk to you about here today is a part of this project where I tested the hypothesis about habitat quality and how it perhaps could affect genetic diversity in a riparian species, specifically this frog. Obviously, genetic diversity is important, and conservation biologists have been concerned with preserving genetic diversity because it is implicated in the fitness of individuals and in the sustainability of a population. So support for this hypothesis would be that subpopulations existing in an unimpacted habitat will have a higher level of genetic diversity than those found in an impacted habitat. Again, the importance of this is that individuals that may be in degraded habitat may have decreased amount of fitness because of this idea of smaller populations being supported by the habitat, experiencing declines. As genetic diversity decreases, the probability of extinction increases. And so this is why I was concerned with trying to determine and assess the level of genetic diversity and see if there is any kind of relationship. There have been very few studies that have looked at this kind of relationship, and there's been nothing on riparian species. So I thought this would be an interesting thing to do. All right. Okay, here we go. Help, please. All right, so to test this, I was looking at a section of the Eel River that runs here at the beginning. Actually, this is part of the Eel River in Mendocino National Forest, and it runs through. This is a man-made reservoir that has a dam here and then goes through this area. So I surveyed 13 creeks along this stretch of the Eel and divided them into two sections. The upper section lies within the Mendocino National Forest, and then the lower section is downstream. Right? So the upper section I consider to be my baseline unimpacted site. And I consider this unimpacted because there are no paved roads in this section of the forest. There's no sort of habitat alteration. Specifically, there are no dams on this part of the river. It is relatively, there's very little human activity. It straddles the Snow Mountain Wilderness region here, so overall it's very high quality habitat. Conversely, the lower section is actually impacted because it is outside the boundary of the federal lands. So there's housing in this area, and there's actually dam structures on this stretch of the river as well. So here are just a list of the potential anthropogenic factors that could be affecting this section of the river. So yesterday you heard all about, and today too, the impact of dams on these frogs potentially, but also the fact that there is paved roadway adjacent does lend itself to increased runoff and increased access by humans. There's also some areas where livestock is adjacent to the streams and the river. And again, there is housing in this region where people have actually PVC conduit running from their house into the streams. Presumably runoff may be directly deposited in the streams. All of this adds up to an alteration of microhabitat. Next slide. All right. So, and again, you can go ahead and click it again. There we go. So on this section, again, just to show you where these dams are, this is the Scott Dam here, and then Lake Pillsbury's behind that. There's also another Cape Horn Dam here, which we see, and it's important to note that there is a diversion tunnel that runs water off of the Eel in this section to go down to the Russian River. Okay. So the field methods included surveying each creek by hand and capturing these, surveying and capturing the frogs by hand, and then taking a GPS reading of the locale, taking measurements of the frogs, and then taking a toe clipping of each. We collected 286 individuals from 13 creeks in the two sections. We then conducted molecular analyses on the samples. And I chose to use two markers because using either one has potential drawbacks. And this is because of the differences in mutation rate, also the difference in effective population size, and mode of inheritance. So as you can see, mitochondrial DNA is passed on from mother to offspring, whereas the rapids, which are randomly amplified polymorphic DNAs, are biparentally inherited. Also, it's important to note that rapids are somewhat cost-effective, much cheaper than, say, microsatellites or even DNA sequencing. So the molecular methods, first of all, we isolated DNA using the QIAGEN minikit. We then amplified the ND2 gene of the mite DNA using primers provided by Amy Lind. And they were then aligned using sequencer software, and distinct haplotype sequences were deposited in GenBank. For the rapid analysis, it's important to note one of the potential drawbacks and the reason that people don't always want to use rapids is this question of reproducibility. We sort of got around that problem in that we were able to use this special kit. It's the Amersham Biosciences Ready to Go Rapid Analysis Beads and Primer Kit. And the neat thing about this is it comes as little dried beads. The PCR reaction is all dehydrated in this nice bead. You just put it in the tube, you just add your DNA, and then off you go. What that does is that reduces human error, pipetting error, and that also optimizes, again, that the conditions are going to be the same, constant, for each PCR amplification. So PCR products were then run out on a 2% agarose gel and then scored. And then a variety of statistical packages were used, Harlequin, DNASP, and GenePop on the data set. This is just a gel photo that shows you what this rapid banding profile looks like. Each gel had a 1 KB and 100 base pair marker run alongside it. And each gel was scored by 3 distinct people. Only those bands that were agreed upon by each individual were kept in the data set. So if a band is present, it would be a 0. If it was, a 1. If it was absent, it would be 0. And so that's how each was scored. We used 3 primers for this, and they were all polymorphic. And you'll see here for the results, they produce these individual haplotypes that could be analyzed. So looking at the overall genetic diversity, the first thing that I did was I grouped all of the individuals together according to the tributaries that they were collected on. Those were considered to be subpopulations. And as you can see from the mite DNA, the 511 base pair region produced 10 polymorphic sites, which resulted in 9 distinct haplotypes. The haplotype diversity was 0.798. This is pretty high. all the substitutions were transitions, and then we thus tested for neutrality to make sure that this was actual neutral variation that we were observing. It was not significant, so it was presumably neutral. The F statistic is a statistic of structuring the amount of differentiation among punitive subpopulations. An FST of 0.06 suggests that there is somewhat high levels of gene flow between creeks. For the rapids, we saw a similar pattern, although it's important to note here that the FST value is a bit higher. And again, this is not totally unexpected because of the differences in the mode of inheritance. Recall that the FST, the rapids effective population size is 4 times greater than that of the mite DNA. It's also a larger number of loci that we're sampling, and so it's a more variable marker. And finally, rapids have a higher mutation rate than mite DNA. So the effects of genetic drift are a little bit more revealable, I should say, by rapids than they are with mite DNA. When I went ahead and ran a phylogenetic analysis, doing a UPGMA, which is a stepwise comparison of the genetic distances, with the rapids data, a structuring was observed. And what we see here, this is the lower section, all the creeks are grouping together, and this is the upper section, the unimpacted region. They are all grouping together. But notice already we're seeing more diversity, greater level diversity from this tree in the upper section than the lower section, forming 2 distinct clades here. When I compared the 2 sections, the upper to the lower, a pattern starts to emerge. And again, this is one of the upper section having higher levels of genetic diversity than the lower section. In all 3 estimates, the number of polymorphic sites was significantly higher in the upper section. The gene diversity also higher, and the among tributary variation higher as well. The same pattern holds for the mite DNA. Again, lower number of haplotypes, lower gene diversity, and lower among tributary variation rates. And these estimates were statistically significant. So what can we conclude from this? Basically, first of all, it's important to note that this section of the Eel River does support a genetically diverse population. And again, the entire section, even the lower section, overall, the diversity is high. And that's important to note. But when we compare the 2 sites, the upper section does exhibit greater diversity. And it's important to note that this section is devoid of human activity for the most part. Again, it's a natural flow regime. There's no dams, whatnot. So this allows for a larger population to be supported. This is not the case for much of Rana boylii's current range, and probably why most of the populations are considered to be threatened. Amy Lynn did her PhD research, and she reported on a study that she did looking at 6 different hydrologic units. The within variation rate she got was between 40 and 70% much lower than what we saw here. So that's important to note. So both the rapid and the mite DNA supports the hypothesis. Lower estimates of genetic diversity and gene flow were observed for the more heavily impacted habitat. And again, so what we're seeing here is that there is an indirect effect on the quality of habitat on the genetic diversity. And obviously, that is an important thing to keep in mind. So for the future work, I hope to continue to sample in this section, but I also hope to sample additional river tributary systems to see if this pattern holds up. I would also like to incorporate microsatellites. I've been working on developing some primers now for a few years, and I'm going to keep at it. And I would also like to incorporate direct observations to see what's actually happening with the dispersal. Finally, I'd like to acknowledge the folks at the California Academy of Sciences, Jeff Wilkinson and Jens Vindum, who supported me in the fact that they show me where to go and catch these guys. They also supplied me with a couple of DNA samples to begin. And then also my students, Joseph Polk, who helped to collect the frogs with me, and my undergraduates that helped score, and the University of San Francisco for providing funding. At this time, I'd be happy to answer any questions. How many other places could I replicate this? I'm not sure, because it's really hard to find the unimpacted area that is contiguous with an impacted area like this. This is sort of a fortuitous situation. But I think it's important to look at more than just one system. It's hard to know if that, it could, you know, that's one of the things. You have to look at other systems to make sure that this isn't just some strange anomaly. Yes, I have looked at the patterns within streams as well as between streams. I've basically looked at it every way you can. Really, the only pattern that holds up is when they actually group as streams. When they're in chunks, it seems that when distances greater than 10 kilometers seem to impede gene flow, and so that's when you start to see structuring. But otherwise, there's really no other patterns that hold up besides that.